Hello, my name is Blake Kruger. I'm a senior chemistry student in the Department of Science. I'm going to be speaking to you today about the research that I've been doing over the last 4 years on a novel Kaposi sarcoma therapy. This presentation is kind of an amalgam of the work that I did and presented at LSU Discover as an LSU Discover scholar, and also the work that I presented at my thesis defense in the fall of 2016 in the Haber lab. So what I'm going to be doing today is I'm going to first give you some background on the epidemiology of Kaposi sarcoma. Then I'm going to speak to you a little bit about second harmonic generation, which is the experimental technique that we use in the Haber laboratory. Then I'm going to give you the information about some prior work that heavily influenced the creation of our therapy. Then I'm going to speak to you a little bit about the work that we did to prove that this phenomenon called plasmonic resonance will amplify the rate at which we can deliver this therapy. And then lastly, I'm going to show you the therapy itself. So, you just heard me say Kaposi sarcoma, and I know the first thing that's kind of rolling through your mind at this point is, well, you said Kaposi sarcoma, so why is there a map of HIV incidence in the United States on the screen right now? And the answer to that is Kaposi sarcoma is not very well studied in the United States. And when you have an HIV-associated illness, meaning if you have an illness that occurs when you have HIV, just as Kaposi sarcoma is, you can use HIV incidence as an analog to look at Kaposi sarcoma incidence, our illness of interest. And so that's what I'm showing you here. And what you can see by this image is that there are 3 places in the United States that are vastly affected by HIV. Those places are New York City, Florida, and Louisiana. And most germane to our discussion in Louisiana are the 7th and 9th wards and just north of LSU itself. So these areas are heavily impacted by HIV. And what I'd like to do now is give you an idea of who is being impacted by these illnesses. And what you can see here is if we focus on the area germane to our discussion, here in the South, 54% of individuals who are dealing with HIV as an illness are African American. And this is true for Kaposi sarcoma. It's been recorded that a majority of people who are dealing with Kaposi sarcoma are young African American males. And these individuals are generally between the ages of 18 and 28. And what's so important about this is that in the South we have the lowest healthcare attainment in terms of this particular demographic. So we have people who are really hurting, who don't have the ability to get healthcare for their illnesses. And Kaposi sarcoma is one of those illnesses that's really swept under the rug. So what we are trying to do is create and deliver a low-cost, low-skill therapy for Kaposi sarcoma that can be easily used in clinics to help treat these individuals who are financially strapped. So what I'm showing you here is really the second to last slide of our Kaposi sarcoma background. So what you see on the left, I'm going to be proving to you that HIV incidence and Kaposi sarcoma incidence are integrally related. So what you see on the left is a map of HIV incidence in Africa. And when I click the next button, this is a map of Kaposi sarcoma incidence in Africa. And what you can see is they superimpose very well with one another. And you might be asking, well, what's the reason for that? And you can see in the center here, you start with an individual who's HIV positive and immunocompromised. These individuals are then co-infected with a very common herpes virus called human herpes virus 8. And then from this co-infection and a decreased immune system, they develop Kaposi sarcoma, which is a soft tissue sarcoma. It's a macular-papular rash, which you can see right here on the right. These individuals have these splotchy red and purple papules that appear on their extremities in the first 2 stages, so on their legs and arms. And then in the 3rd stage, this cancer moves to visceral involvement, so it begins to involve your organs, and at that point, mortality rates are very, very high. So what's so exciting is there have been new advances in Kaposi sarcoma biology that we can now take advantage of in order to treat Kaposi sarcoma. So the Parsons group at the LSU Health Sciences Center actually has done a lot of research on this, and so what they've done, you can see here depicted in this image. You can see that extracellular matrix metalloproteinase, or EMPRIN, these red transmembrane proteins, are integrally involved with Kaposi sarcoma cell migration, invasion, chemotherapeutic resistance, and cell survival. But most importantly, EMPRIN is involved with cellular angiogenesis. And so Kaposi sarcoma, as a cancer, is what's called very angiogenically active, and that means it recruits lots of blood vessels to it in order to supply it with enough resources in order to be biologically active. So if you have a cancer cell that's constantly dividing, it's going to want more and more resources so it can keep dividing at that same rate. So it stands to reason that if you take away the ability, or at least decrease the ability, for this cancer cell in order to divide and to make blood vessels for itself, you should then be able to treat the cancer fairly effectively. And that's just what we've done. We used what's called a silencing RNA, which degrades messenger RNAs from producing extracellular matrix metalloproteinase. So this decreased concentration of EMPRIN in the transmembrane area thus decreases the angiogenic properties of the cancer and therefore will help treat it. So at this point I'm going to speak to you a little bit about the experimental technique that we use in the Haber laboratory called second harmonic generation, or SHG. And SHG is an experimental technique that uses a fancy laser setup that you can see on the far right here that I won't go into. But what is important is this image here in the center. And what you can see, these two red lines symbolize photons, and they're of some frequency, any color of light you want. And when these two colors of light interact with a colloidal nanoparticle sample, they generate a third photon of light of frequency 2 omega. And by monitoring the energy and the frequencies of these photons, you can tell what's happening at the surface of nanoparticles using this particular technique. So what's so important to us is we use second harmonic generation to watch the release of what are called oligonucleotides, which are our RNAs, from the surface of nanoparticles using this technique. And so when you see these graphs later, you're going to see high SHG intensity, so a large number of SHG intensity counts, what they're called. They'll be moving to lower numbers. And when you see this, it means that oligonucleotides are being released from the surface at that point. So the prior research that really influenced us in order to try and make this therapy was first some research that was done in the Haber lab. And what you see on the left is a little bit of research that was done by Raju Kumal. And he's a PhD candidate in our lab, and what he's done is he used gold nanoparticles to attach these RNAs, these oligonucleotides, as I will continue to refer to them. He attached these oligonucleotides to the surface of nanoparticles using a specific linker that will break when you shoot a certain color of light or a certain frequency of light at it. And those are called photolabile bonds, the bonds that break at a certain frequency of incident light hitting it. And he did these investigations, and the linker he used was a nitrobenzyl photocleavable linker, which is a very good linker, but it lyses at 365 nanometers. And if we look at a color spectrum, 365 is in the ultraviolet, is in the low visible to ultraviolet. And what's so important about this is you might say to yourself, well, I've heard of that before. That's when the sun shines down and I'm on the beach and I have to put on suntan lotion to protect myself. Isn't that right? And that is right. Ultraviolet light is actually very bad for therapeutic purposes. And the reason for that is not for, you might be thinking you'll develop skin cancer. However, this ultraviolet light is a bad therapeutic modality because it's absorbed by melanin in your skin. So your skin absorbs this UV light, which prevents it from reaching the therapy, which will prevent it from being released. And so taking this into account, we thought, well, what is a type of light that's really well transferred through the skin? And the answer is infrared light. And there was actually a physician who has done some similar research to this that has done it before. His name's Anil Sood, and he works out of the Texas system. And what he did was he conjugated this chemotherapy called doxorubicin to the surface of hollow gold nanoparticles. And when he did, he gave these to breast cancer patients. It reached stage 2 clinical trials, and I believe it's still there. It's awaiting stage 3 confirmation. And they were wildly successful. So taking these two ideas and the new biologic developments that we took from Kaposi sarcoma, we can now start to make a little picture. Well, we need some nanoparticles that will allow us. We need some nanoparticles in a photolabile bond that will let us lyse in the near infrared, and we need a silencing RNA to deliver. So before we got to making the therapy, we did some other research, and this other research required us to synthesize gold and silver nanoparticles. And what we were doing is we were looking at the effect of what's called plasmonic resonance with the release of oligonucleotides. And the plasmon, you can think about it being at a wave pool at a big park. You have a bunch of people in floaty tubes, and they're all at the top of the water, and they're floating with the waves. If you replace those people with electrons, that oscillation is what's occurring at the surface of nanoparticles when they're at this particular plasmonic resonance frequency. So what we did was, when we synthesize these nanoparticles, we also characterize them. We characterize them using transmission electron microscopy and a Curie 500 UV vis spectrophotometer. And from that, we gathered these TEM images at the top and these extinction spectra at the bottom. These TEM images show us what we've made, and the extinction spectra at the bottom show us what this frequency of plasmonic resonance is, where the most electrons are oscillating at what frequency. And so what you can see here is we've adequately characterized these nanoparticles that we synthesize using the citrate method, these gold nanoparticles. We did the same with silver nanoparticles, and you can see again the TEM image here and the extinction spectra below. And we use the same nitrobenzyl photocleavable linker. And you can see on the left what we were going for. We were looking to release this oligonucleotide from the surface using this NBPC, or nitrobenzyl photocleavable linker. So we did second harmonic generation experiments on this. And what we did was we proved here on the left that our oligonucleotide, or our RNAs, as I will refer to them now, our oligonucleotides were released only when they were irradiated with 365 nanometers, which means that the complex that we made is selective for 365 nanometers of light. So it'll only release the oligonucleotide when we want it. What we see on the right here is what's called a wavelength dependent study that we did. And in this wavelength dependent study, we looked at what wavelengths of light would actually release these nanoparticles, release these oligonucleotides from these nanoparticles. And what we found was really interesting. There were a number of frequencies that did, but there were certain frequencies in particular that were the best at it. And you can see with this decrease here, the lowest line that occurs at the shortest time, is showing that there was the most released in the most oligonucleotide released in the shortest amount of time. And when we compared different nanoparticles with their oligonucleotides that were released, what we ended up finding was that when you have a nanoparticle system whose plasmon resonance, the most electrons that are found at the surface at a certain frequency, when that plasmon resonance light color or frequency is the same and in resonance with the photolabile bond, or the releasing bond that we release our oligonucleotides with, you see the maximum rate of release, and that's just what we saw. We saw a maximum rate of release at least fourfold in silver nanoparticles as compared to polystyrene sulfonate nanoparticles, which was our control group. So at this point, we took all these different ideas and we brought them together. And using a synthesis paradigm for gold, silver, gold core shell nanoparticles that was first developed by Tony Karam here in the Haber group, we created a nanoparticle whose plasmonic resonance was at 800 nanometers. We replaced the nitrobenzyl photocleavable linker with what's called a retro-Diels-Alder linker, which uses the same concept. It's a photolabile bond, but it lyses at 800 nanometers, and you can think 800 nanometers is the color red, and that's in the near IR, and that transmits well through the skin, so it's good for therapeutic purposes. And since there's gold on the surface of these nanoparticles should not be cytotoxic. As well, we took the sRNA for emprin to downregulate emprin in these cells, and we conjugated it as well. And so what you can see here is a synthesis paradigm. We took the citrate and the silver nitrate synthesis methods, and we just alternated them. We used this sodium citrate gold nanoparticle synthesis to make the seed, then layered it with silver nitrate, then layered it with gold once again. We then functionalized these nanoparticles using what's called a salt aging technique, and then we created this therapy. So this image here in the center is showing you the retro-Diels-Alder linker that we have used. And it's really interesting because when you shoot it with light, it kind of unfolds and then releases. So this is just a more in-depth depiction of that. This image is that second step in the synthesis process. You saw the first step, the gold seeds. This is the silver coating on the gold seeds here. And you can see that the plasmonic resonance is shifting. And these are the gold, silver, gold core shell nanoparticles, the last step in that. So this is what we used for the therapy. And what you can see by this plasmonic resonance here is that we've shifted the plasmon all the way to 795 nanometers, which is almost 800. And our retro-Diels-Alder linker lyses at 800 nanometers. So we took these functionalized nanoparticles, and we performed two SHG experiments. The first, we did second harmonic generation on blank core shell shells, so there was no functionalization. And as you would expect, we didn't see any appreciable change of second harmonic generation over a very long period of time. And then we did a second harmonic generation experiment where we used 800 nanometers and we compared core shell with the nitrobenzyl photocleavable linker to the core shell with the retro-Diels-Alder linker. And this light is incoming at 800 nanometers. So what we see is this complex is selective for 800 nanometers, and we see very fast rates of release. What's so important about this is we saw release in 41.2 seconds. And that's incredible. When you think about taking an aspirin, it'll take you 10 or 15 minutes to get relief for your headache or for your joint pain. But you can have a patient come in, sit down, tourniquet their leg to restrict blood flow, inject just under the skin with a regular needle this therapy, then take an IR lamp source, put it over their leg, irradiate the leg, remove the lamp source after a minute, slowly untourniquet the leg, and then send them on their way. They'd be out of the office in 15 minutes in a low-cost, low-skill therapy for traditionally underrepresented and minority groups. So this brings us to the discussions, conclusions, and some future investigations. So what I've showed to you today in this presentation is that the prior research that we did with our silver nanoparticles proved that we can amplify rates of release of our drugs from the surface if we make our release linker in resonance with the plasmon of the drug delivery modality. Then I showed you how we synthesized, characterized, functionalized, and tested these gold, silver, gold core shell nanoparticles as a novel Kaposi sarcoma therapy. And at this point, we now need to make clear that more investigations are needed. We need to continue compiling reproducible data for these siRNA releases. we need to also determine the cytotoxicity in a numerical way for these gold, silver, gold core shell nanoparticles in order to test if they're safe for patients. And then lastly, we need to quantify the concentration of siRNA that is released per nanoparticle when it's irradiated with 800 nanometers of light using fluorescence microscopy. And we also came up with an interesting side project that we could do, and that was looking at whether or not the spiky artifacts that you saw on the gold, silver, gold core shell nanoparticles amplified second harmonic generation intensities at the surface, kind of acting as small antennae. And what you see at the bottom is simply a summary of all the nanoparticles that I've created in this. So at this point, I would like to acknowledge my thesis committee, Dr. Granger Babcock, Dr. Gracia Vicente, Dr. Lewis Haber. I'd also like to thank the Ogden Honors Leaders Program, who financially supported this endeavor. I'd also like to thank the LSU Discover's Discover Day program for naming me an LSU Discover Scholar. I'd like to thank all of the other individuals who helped us along the way, our collaborators and my group mates. Some highlights: Dr. Parsons, Dr. Plaisance Bonstaff, Dr. Elzayed, Dr. Heza, Dr. Parak, Mr. Kamal Smith, Corey, Taylor, Dicky Kumbra, Dr. Cueto, Dr. McCarley, and Dr. Shaw for letting me use the microscopy labs here at LSU, and our very close collaborators, Dr. Daniel Hayes and Mr. Mohammed Abu Laban, Dr. Tony Karam, who helped us with the synthesis of the core shell, and Ms. Seegers and Dr. Jonathan Earl, as well as my family for their continued academic and financial support. Thank you.